Well, good afternoon, and thank you for the privilege of introducing Talaris Therapeutics to you. As you see from the little logo in the bottom left-hand corner, we were recently honored to win Xconomy's Big Idea of 2019 award at a recent gala in Boston, and so I'd like to fill you in on why it's a big idea. So briefly, we have the audacious goal of changing solid organ transplant and, more importantly, the lives of patients receiving solid organ transplants and potentially also severe autoimmune diseases. But I'll start with a little bit of background, the problem we're trying to address. As all of you know, organ transplant is life-saving, but the irony is that the drugs that are needed to avoid the recipient rejecting the organ are themselves inherently toxic to the kidney. And as a result, the transplanted kidney tends to only have a 12 to 15-year life, sometimes longer, sometimes shorter, and eventually a younger recipient will need a new transplant, maybe even more than one in their life. On top of that, there's a high pill burden, and there's a need to treat all the comorbidities associated with taking lifelong immunosuppressives. These include a number of cardiovascular comorbidities, which actually result in cardiovascular issues being the leading cause of post-transplant mortality in transplant recipients rather than failure of the transplant graft. Tremendous quality of life impact in terms of sleep disturbance, CMS, depression. You're, of course, chronically immunosuppressed, so you are at risk of serious infection, and you have a much higher risk of certain types of cancers. So not a pretty list, but it's better than being dead, which is the current standard of care if you don't receive these drugs. So what we're trying to do is we're using our cell therapy, which is FCR001. It's essentially a personalized single-dose cell therapy, which we derive from the stem cells and other immune cells of the organ donor a few weeks prior to the transplant. We administer these to the transplant recipient with a non-myeloablative conditioning regimen. And the goal is to get these stem cells from the donor to engraft into the transplant recipient and, in essence, enable the transplant recipient's immune system to now recognize the donated organ as self. And that allows us over time, and the clinician over time, to wean the transplant recipient off all immunosuppression. Our lead indication is in living donor kidney transplant. And our goal in that case is to allow them to be durably weaned off all immunosuppression, typically within 12 months of their transplant. I'll share with you some pretty compelling phase 2 data that we've announced recently, and we're about to be starting a phase 3 trial later this year. But we believe that, in fact, there are significant applications well beyond living donor kidney transplant. This is, in fact, a pipeline and a product. So a few highlights from our phase 2 study. This was a phase 2 study conducted between 2009 and 2016 at 2 leading transplant centers in the United States. We enrolled 37 living donor kidney transplant patients. And as you can see, 70% of those patients we were able to wean off all immunosuppression, typically within 12 months of their transplant. And in fact, once we sort of locked down all key parameters of our protocol and our processes of the last 17 patients that were enrolled in this open label trial, we were able to get 14 of those 17 off all immunosuppression. Quite remarkably, every single one of the patients that we've been able to get off immunosuppression has stayed off immunosuppression. We have followed these patients for a median of 5 years from the time of their transplant, and the earliest of these out to 10 years. And another interesting finding, provocative enough that we're going to be building a phase 2 around it, is that a subset of the patients in this phase 2 study suffered from a kidney-related autoimmune disorder. And as many of those in the audience will know, even if you've had a transplant of a new kidney, if you had a kidney-related autoimmune disorder, there is a very good likelihood that it will return and attack the new kidney, anywhere from 25 to 60% of cases. And somewhat provocatively, it's a small number, but every single one of the patients that we were able to tolerize and get off immunosuppression, we did not see a single one of them who had recurrence of their autoimmune disease. One of the other things that's quite remarkable, I would say even unprecedented, about this data set is that we achieved equally robust results across all degrees of HLA mismatch between the donor and the recipient. And as you can see from this bar chart, which shows the distribution of recipients and the degree of HLA match between the recipient and the donor, almost 3/4 of the recipients that we successfully weaned off all immunosuppression had an HLA match of 3 or less between the donor and the recipient. So we do not require that there be a high degree of HLA match, and as a result, if you're an eligible kidney donor, odds are that you're going to be an eligible stem cell donor for purposes of our trial. We also saw during the phase 2 a very good safety profile, notwithstanding the fact that the vast majority of the patients in our trial had high degrees of HLA mismatch. We saw comparable graft and patient survival compared to the standard of care kidney transplant. Overall, we saw a safety profile that was entirely consistent with standard of care kidney transplant and with HLA matched stem cell transplant. Reminder that we only use a non-myeloablative conditioning regimen here as opposed to a fully myeloablative conditioning regimen. Typically after 12 months, by which time those who were successfully weaned off all immunosuppression, we saw a significant reduction in their AEs since they were no longer on immunosuppressive agents. And very importantly, we did not see any instances of acute rejection or donor-specific antibodies in patients off all immunosuppression. And as I mentioned before, we followed these patients a median of 5 years and out to 10 years. The first 20 patients in the trial have passed the five-year follow-up mark. And a couple of other things that we have observed, which all point to long-term clinical benefits of our therapy, include an improved quality of life. We recently presented some of these data at ATC. Also, better kidney function. As I mentioned at the outset, the drugs themselves are inherently toxic to the kidney. So the hypothesis would be if you would remove the insult from the drugs within 12 months after the transplant, you would hope to see stabilization of kidney function. What we saw in the first 20 patients is essentially no significant difference in kidney function from the time immediately post-transplant to 5 years out, whereas in a cohort of 132 other living donor kidney transplant recipients who were transplanted around the same time at the same center, on average you saw an almost 30% decline in kidney function because they were still on standard of care immunosuppressants. We also saw exactly what we would have suspected, which is an improved cardiovascular risk profile, dramatically lower dependence on hypertensive medication or medications for high cholesterol in our Talaris patient subset than in the standard of care patients that were concurrently treated at the same center. So what is the secret sauce? Well, there are several ingredients, but the most critical ingredient was an invention of Dr. Suzanne Ildstadt, our chief scientific officer and founder, who over 30 years of devoting her life to understanding mechanisms for promoting immune tolerance, discovered a unique type of cell called the facilitating cell. These are non-stem cells. They're CD8 positive, T cell receptor negative, and they perform several critical functions and are a critical component of the product that we're transplanting into the recipient. Namely, they upregulate a number of signals and pathways that promote immune tolerance, including, we believe, BREG. They actively promote stem cell engraftment in the recipient's bone marrow, and they appear to prevent graft versus host disease, which is, of course, something one always fears if one's doing an HLA mismatched allogeneic stem cell transplant. Our lead product is going to benefit from an accelerated and de-risked development pathway. We've received both Orphan Drug Designation as well as RMAT designation from the FDA on the strength of our phase 2 data. We've agreed upon the pivotal study design and endpoints, and we have a single phase 3 that we expect to start later this year, which we believe should be able to support accelerated approval. One of the beauties of our trial is that we've also identified a surrogate marker, typically measured at 6 months, which is very highly predictive of long-term success. It's the same marker that clinicians look to when treating our patients, which is at 6 months, if the organ transplant recipient is demonstrating that more than 50% of his or her T cells and red blood cells appear to be donor-derived, then that's evidence that the stem cell transplant has engrafted and that you have a donor-derived immune system which is active in the recipient. And when we see that, that's the trigger that clinicians use to start down titrating the immunosuppressants over the next 6 months. And as I mentioned before, every single patient that we've been able to take off immunosuppression has stayed off immunosuppression. So in essence, chimerism at 6 months has been an excellent long-term predictor of outcomes. As I mentioned, we're about to kick off a single registration study, which we've named Freedom 1. It's going to be in 120 adult living donor kidney transplant patients. It's going to be randomized 2-to-1 to FCR001 or standard of care. It's an open label trial, and the primary endpoint of that study is the proportion of patients receiving our therapy who are free from immunosuppression and have not experienced a graft rejection event at 24 months post-transplant. We anticipate that we'll be in about 15 sites in the United States, the first of which is Northwestern, and that we'll be initiating this study this quarter. One of the other things, a number of other folks this week have been pointing out the importance of controlling your own cell processing. We do all of the processing ourselves at our cell processing facility in Louisville, Kentucky. We have a relatively simple process. We're not introducing or using any viral vectors. We're not doing any genetic engineering. We're not doing any expansion or any transductions. And we own and fully control all aspects of the cell facility. We lock down all of those processes and controls in anticipation of phase 3. And I'm also pleased to say that we have some pretty broad issued IP claims around both the product and the processes. I mentioned earlier that we believe this is actually a pipeline in a product. Using exactly the same processes, essentially harvesting cells from a healthy donor, we not only believe that we can address living donor kidney transplant, but we also believe that this has the potential for treatment of severe autoimmune disease, where it has already been shown that bone marrow transplant can be curative, but this frequently requires a fully myeloablative conditioning regimen. Our vision is to use a non-myeloablative conditioning regimen with donated stem cells from a healthy donor as a means of essentially re-inducing tolerance in these individuals. And we're also going to be running another phase 2 in what we call the delayed tolerance setting, which is people who've previously received a living donor kidney transplant, where we can go back to the healthy donor, receive a donation of stem cells, and see if we can induce tolerance after the fact, which would open up some of the prevalent population and also potentially open up the entire deceased donor organ transplant space. So we see a real pipeline opportunity here, and all of these will be essentially done with the exact same processes and same product as we use for our lead indication. The company is very well financed. The company raised or announced $100 million financing in April of this year, led by Blackstone Life Sciences and with Longitude and Qiming, very knowledgeable and experienced life sciences investors. We have a terrific set of independent board members and a very experienced management team, including Dr. Ildstad, our CSO and co-founder, our Chief Medical Officer, Nancy Krieger, transplant surgeon by training. I'm the CEO, and a brief anecdote about me. I was leading the diligence into this opportunity while I was an investor, and I found it so compelling that I left my fund, which became Blackstone, in order to join Talaris. So in summary, we're a late-stage cell therapy company, and we have the audacious goal of doing nothing less than revolutionizing the organ transplant space. We are developing our lead product, FCR001, initially for living donor kidney transplant. On the basis of pretty compelling phase 2 data, we're moving now into our registration study, which we believe has a de-risked and accelerated development path and significant pipeline potential. And we have a terrific board and management team, and we control all of the manufacturing and 100% of the rights to the product in all geographies and all indications. I'll just wrap up with the most important thing, the reason we're all here. We're all about the patients. This is a photo of Robert Waddell and his family. He was our first successful transplant recipient back in 2009. And the photo you see on the left is him with his family about a year after he ceased immunosuppression. And he and his family and the kids all grown up recently joined us earlier this year when we announced the financing of Talaris. And he's been off immunosuppression now for almost 10 years. And to quote him, he feels like he never had a kidney transplant. We obviously hope that all of our patients would have those kind of outcomes, but that's what we're committed to. And I thank you for listening to our story, and thank you for your interest.